So students, in previous class we have learned about Mendelian inheritance. Now we will see what are the modifications of this Mendelian genetics. So that in this lecture we will study about types of gene action and modification of Mendelian ratios. Means what kind of gene actions are there and what are the modifications of Mendelian ratio. So first we will discuss about gene interaction. So there are two types of gene interaction. One is intra-allelic and other is inter-allelic. Intra-allelic means within alleles of a gene at the same locus, and inter-allelic means interaction between alleles of different genes which are not alleles. So intra-allelic interaction are complete dominance, incomplete dominance, codominance and overdominance. So first of all we will study about complete dominance. As we have studied in Mendelian inheritance what is dominance and what is recessiveness. So the complete dominance, it states that a type of gene action in which one allele at a locus completely masks the effect of another allele at the same locus. At the allelic pairs that Mendel studied showed complete dominance. Actually Mendel was a bit lucky in conducting his experiments, as he could not get linkage, otherwise he would have different kind of results what he had. So all the allelic pairs that Mendel studied showed complete dominance or complete dominant recessive relationship. That is, one allele at a locus is dominant to other allele. What is incomplete dominance? As its name suggests, the dominance is not complete. That is, incomplete dominance or partial dominance is shown when the dominant allele is not completely dominant to the recessive allele. The heterozygote phenotype is closer to the dominant phenotype than to the recessive phenotype, but is not identical to the dominant phenotype. As we have learned in complete dominance, that heterozygous individual, they were similar to dominant parents, but here that heterozygous individual will be closer to dominant phenotype, but not the exact copy of dominant phenotype. So that plumage color in chicken is a good example of incomplete dominance. Crosses between a true breeding black strain and a true breeding white strain gives F1 progeny with bluish gray plumage. So we will see it here. When in parental generation we cross a female with black plumage with a male of white plumage, they will be having genotype of CB and CW. Then we will have gametes of CB and all CW. In F1 genotype, and in F1 progeny, we will have a individual with CB-CW genotype. So their phenotype will be bluish gray due to incomplete dominance. When in F2, when we allow to mate these F1 individuals, that is bluish gray female mated with bluish gray male, then in F2 generation we will have these kind of genotypes, that is CB-CB, CB-CW and CW-CW. So we will have a phenotypic ratio of 1:2:1, but in complete dominance it was 3:1. So due to incomplete dominance we are having a new phenotype, that is bluish gray. So same thing is discussed here. In parent 1 and 2 we can see black plumage female and white plumaged male, they are crossed. Then in F1 generation we could get only CB-CW, those all were bluish gray in phenotypic appearance. So same thing is discussed here. In this picture we can see the parent 1 and parent 2, the parent one, female black plumaged female is crossed with a white plumaged male. So in F1 generation, we will have a bluish gray plumaged individual. That is due to incomplete dominance. But when we allow these bluish gray male and female of F1 to cross, then in F2 generation we will have these three genotypes, and they will represent three different phenotypes. So instead of getting a phenotypic ratio of 3:1, we are getting a phenotypic ratio of 1:2:1. Okay, that is due to incomplete dominance. In plants, incomplete dominance is shown by a plant, the snapdragon. What is snapdragon? A cross of red flowered variety represented by CRCR with a white flowered CWCW produces a heterozygote of CRCW F1 plant with a pink flower. So one parent is red flowered, another parent is white flowered, and in F1 we are getting a pink flowered plant. So the interbreeding, the F1 produces an F2 with a 1:2:1 ratio of red, pink and white. Same as we discussed in plumage example, here also we are having three phenotypes and three genotypes. So instead of getting 3:1 of phenotypic ratio, we are getting here 1:2:1 of phenotypic ratio. Same thing is discussed here. So here we can see the red colored plant, the red flowered plant, and that is white colored flower plant. And in F1 generation, we are getting a pink flowered plant, a plant having pink flowered. In F2, we are getting three types of genotypes and three types of phenotypes: red, pink, and white in 1:2:1 ratio. What is codominance? Now we'll see what is codominance. As its name suggests, codominance, both alleles of a pair are fully expressed in a heterozygote individual. That is, in a heterozygote individual, instead of expression of one allele, there will be expression of two alleles. So the F1 heterozygote exhibits the phenotypes of both the parents, both the homozygous parents. So there is no dominant or recessive allele. Both alleles contribute equally to the production of phenotype. So in codominance, both the alleles will contribute to the phenotype of that individual. The inheritance of red, white and roan color in shorthorn cattle is the example of this codominance. The R allele leads to production of red pigment in the coat hairs, while the W allele leads to no pigment formation. So that R allele will lead to red pigment hairs, while when W allele is present, it will lead to no pigment formation. That is, it will be a white colored hair. In animals with capital R genotype, all the hairs are red. And with WW genotype, the appearance of hair are white. In heterozygous animals, that is capital R W, the appearance of these pigmented hairs and non-pigmented hairs will be together. So that will give a appearance of roan color. Means white and red pigmented hairs will be there, but that phenotypic expression, appearance will have a roan. Same thing is discussed here. When a female of white hair and a male with red hair are crossed, then in F1 generation, we will have all roan. So instead of getting red or white hair, we are having both red and white in same individuals. That will give an expression of roan. So another example of this codominance is ABO blood group system in humans. So that heterozygous individual IA, IB will have both antigens A and B. So these are codominant. So the allele IA and IB are codominant. Means in heterozygous individuals, we will have both the alleles together, IA and IB. Overdominance, what it is? As its name suggests, overdominance means the expression of or performance of heterozygotes or hybrids will be over and above of both the parents. So the type of gene action in which the performance or phenotypic expression of heterozygote animals is more extreme than that of both homozygous parents. Example of this is sickle cell anemia in humans. So here we can see the genotype HBA, HBA, then HBS, HBS and HBA, HBS. So the survival rate of HBA, HBA is high than HBS, HBS. Then so here we can see the heterozygote individual HBA, HBS is having the survival rate of maximum survival rate, as we know, due to this overdominance phenomena. These individuals with HBA, HBS will have the highest survival rate. So there are two types of gene interaction. First is multiple allele control the same trait and their interaction produces a new phenotype. So multiple alleles will be there, but when they are together, due to their interaction, a new phenotype will appear. And second one is epistasis. One or more genes mask the expression of other genes and alter the phenotype at different locus. So gene interaction that produces a new phenotype. Different genes control the same trait and collectively produce a new phenotype. For this example is comb shape in chicken. In chicken, 4 phenotypes resulting from dominant recessive alleles at 2 loci. So here we can see that for a rose comb to present, that genotype should be capital R dash small p. For a pea comb, small r, capital P dash. And for a walnut, that is both the dominant alleles, that is capital R and capital P, are present. Due to their interaction, the new comb is appeared here, that is walnut, that will have a genotype of capital R dash, capital P dash. And for single comb, it should be a homozygous recessive for both the alleles. So the cross true breeding rose combed and a pea combed chicken. So when we will cross a true rose combed and a true pea combed chicken, what we will get? We can see. So the interaction of two dominant alleles, that is capital R and capital P, produce a third phenotype, walnut, of F1. So when we will cross a when we will cross a rose combed individual or male or female to a pea combed male or female, then in F1 we will have all walnut. Why we are getting these all walnut instead of getting red pea or rose comb? Because these alleles, capital R and capital P, they interact with each other and produced a new phenotype, that is a walnut comb. Fourth phenotype, single comb, appears in F2 only. In F2, the ratio of walnut to rose to P to single will be 9:3:3:1, and it fits in Mendelian ratios. Multiple genes involved and interaction of two dominant alleles, capital R and capital P, produce a factor that modify comb shape from a single to more complex form to a walnut. So these are the pictures, these are the photographs of these combs. This is in A, you can see red comb, sorry, rose comb is there. then walnut, then pea, and then single comb. Same thing has been discussed here. So here we can see when a female of rose comb is crossed with a male of pea comb, having pea comb, then in F1 generation we will get only walnut comb progenies. This is due to interaction of these capital R and capital P alleles. And in F2, as we have seen already, we will get a 9:3:3:1 ratio of walnut, rose, pea, and single comb progenies. Epistasis, what it is? Epistasis is a Greek word meaning standing over. It was first used in 1909 by Batson to describe a masking effect, an interaction between a pair of loci in which the phenotypic effect of one locus depend on the genotype at the second locus. That is, it is the interaction between the alleles which belong to different genes at a different locus. So genes whose phenotype are expressed is epistatic and whose phenotype is not expressed or altered, that is hypostatic. Epistasis is a form of gene interaction in which an allele at one locus interferes or masks the effect of another allele at a different locus. A gene that masks the expression of another allele or genes is called as epistatic, and whose expression is suppressed or altered, that is said to be hypostatic. In F2, the number of phenotype classes are less than 4. So there is a difference between dominance and epistasis. So what is the difference between dominance and epistasis? Dominance involves intra-allelic gene interaction and epistasis involves inter-allelic interaction. Dominance is a phenomena which is due to interaction or which is intra-allelic, that is alleles of a same gene. And epistasis is a phenomena which involves inter-allelic interaction, that is interaction between non-allelic genes. One allele hides the effect of other allele at the same gene pair or at same loci, but in epistasis that gene pair will be at different loci. So the common epistatic ratios are dominant epistasis that will have 12:3:1 ratio, recessive epistasis that will have 9:3:4 ratio, duplicate gene with cumulative effect that will have 9:6:1 ratio, then duplicate dominant gene that will have 15:1 ratio, duplicate recessive gene that will have 9:7 ratio, then dominant recessive interaction that is 13:3 ratio. We will discuss them in detail. So first we will discuss about dominant epistasis. What it is? It is caused by the dominant allele of one gene masking the action of either allele of other gene. Suppose two genes are there, the dominant allele of one gene will mask the action of alleles of other gene. So in this dominant epistasis we will have a ratio of 12:3:1. Instead of getting a ratio of 9:3:3:1, we will have a ratio of 12:3:1. So for this type of epistasis, example studied in summer squash, Cucurbita pepo, a common fruit in which white, yellow and green colors are there. White is dominant over colored squash, that is white will be dominant over colored squash, and in colored squash yellow is dominant over green. So in this picture we can see when we will cross a white squash with a green squash having a genotype of capital A, capital A, capital B, capital B, and small a, small and small b, small b. So we will have a gamete capital A, capital B from white and small a, small b from green. So when we will cross them in F1 we will have all white having a genotype of capital A, capital B, small b. And when we allow these F1 to cross each other, then in F2 we will have a ratio of 12:3:1. That is, white will be 12, yellow will be 3 and green will be 1. Why it is? Because here we can see for a squares to be white, that capital A dash, capital B dash, genotype must be there. But when this small a, that is another allele of A gene is there with other alleles of B gene, still we will have a white phenotype. So instead of getting a 9:3:3:1 ratio, we got 12:3:1 ratio in F2 generation. So our next one is recessive epistasis. In this we will have a 9:3:4 ratio instead of 9:3:3:1 ratio. It is also called as supplementary interaction. Two genes involved in coat color determination. Gene B determines whether black or brown pigment is produced. So this example we will study in golden retriever breed of a dog. So in golden retriever, two genes involved in coat color determination. One is B gene, capital B gene, and other is small b gene. So capital B determines whether black or brown pigment is there, and another gene is capital E, which determines if pigment is deposited in hair, it will be golden retriever, that is small b, make either black B dash or brown BB pigment at nose but not in fur. The recessive allele is epistatic to other genes when homozygous, hence the name recessive epistasis is given. Why recessive epistasis is given? Because the recessive allele is epistatic to other genes. Coat color inheritance Labrador retriever, it's a breed of dog. When in parental generation we take a golden retriever and cross with a black, that in F1 we are having all black progeny. Then when we made these F1 male to F1 female, then in F2 we will have a ratio of 9:3:4. That is 9 black, 3 brown and 4 golden color, golden retriever dogs. Then dominant recessive interaction, here we will have a ratio of 13:3 instead of 9:3:3:1. Here in this phenomena, the dominant gene at one locus and recessive homozygote at other locus produce the same phenotype effect. What it is? The dominant gene at one locus and recessive homozygous at other locus will produce a same phenotype. That is capital A dash, B dash and small a, capital B dash and small a, b will produce the same phenotype. Example is plumage color in chicken. So here we can see when we cross a white leghorn with a white Vendot, then in F1 we will have all white progeny. But when we made this F1 male, F1 female, then in F2 progeny we will have a ratio of 13:3. That is 13 we will have white and 3 we will have colored. Why it has happened? Because as we can see here, the capital A dash, capital B dash, it's producing white. Then this small a, when it is present with alleles of other gene, then it's producing white phenotype. So instead of getting a 9:3:3:1 ratio, we are getting a 13:3, that is 13 white and 3 colored progenies in F2 generation. So next one is duplicate gene with cumulative effect. Here we will have a ratio of 9:6:1 instead of 9:3:3:1. Dominant condition at either locus produce the same phenotype. Dominant condition at either locus will produce the same phenotype. That is, the dominant alleles of either gene will produce same phenotype. Both the dominant non-allelic alleles when present together give a new phenotype, but when allowed to express independently, they give their own phenotypic expression separately. That is, when both the dominant alleles are together, they will interact and produce a different or a new phenotype. But when they are allowed to express their original phenotypic expression, then they will produce their own phenotype. In the absence of any dominant allele, the recessive allele is expressed. So, it's an example for this type of epistasis is, in pig, capital S and small s are allelic genes. Capital S gives sandy color and small s gives white color. In non-allelic gene, capital R also gives sandy color. But when both the dominant genes interact together, they give red color. That is, when capital R and capital S are together, they will give red color. But when they are present separately, then they will produce sandy color. But okay, then non-allelic gene does not interact with smallest. So here we can see when the sandy color male or female is crossed with sandy male or female, then in F1 we are getting red colored progeny. Instead of getting sandy colored progeny, we are getting red colored. Why we are getting red colored? Because of interaction between capital S and capital R genes, which are non-allelic. So here we can see these are the male gametes and female gametes. Then out of these we are getting 9 red colored due to interaction between capital R and capital S. Then 6 sandy color, as we can see here, when the capital S is present individually and capital R is present individually, then they are producing sandy color. But when they are present together, they are producing red color. And when smallest is present, then we are getting white color. Next one is duplicate a dominant gene. Here we will get 15:1 ratio. Each dominant locus produces the same phenotype effect and there is no cumulative effect. That is, at least one of the dominant alleles is necessary for the phenotypic effect. That is, either capital A or capital B is present, then they will give same phenotype. The duplicate genes are also called as pseudo alleles. That is, capital A and capital B, they are called as pseudo alleles because they are giving the same phenotypic expression when present together or separately. Example for this type of epistasis is the capsule shape of Shepherd's spurs, that is capsella, occur in 2 different shapes, that is triangular and top shape. When we cross a triangular shaped fruit with a top shaped fruit, then we will have in F1 all triangular, but when we allow these F1 progenies to mate together or for self-pollination, then in F2 we will have 15 of triangular and 1 is top shaped. Why it is? Because capital A and capital B are producing same phenotype, whether they are present together or present separately. Then duplicate recessive epistasis. Here where we will get 9:7 ratio. Either recessive homozygous suppresses the expression of other locus. That is, recessive homozygous suppresses the expression of other locus, also known as complementary gene action. For example, for this is flower color in P, that is Lathyrus odoratus, we all know. In that we will have capital A dash capital B for purple color, and for white it should have capital A dash small b small a capital B dash and small a small b genotype to be a white colored flower. Here we can see 2 plants are mated which are white colored, which are having white colored flower. When they are mated, in F1 we will have all purple colored flowered. But when the self-pollination is allowed or permitted in these F1 progenies, then in F2 we will have 9:7 ratio of purple and white flowered plant. Thank you, students.